like to extend a very warm welcome to all of the participants for joining our research talk today. This talk is part of the research talk series organized by Research Center for Pharmaceutical Ingredients and Traditional Medicines, Research Organization of Health, National Research and Innovation Agency, BRIN. My name is Marisa Angelina, and I'm very pleased to be a master of ceremony for these talks. Ladies and gentlemen, to begin this program, we are pleased to invite Professor Yuli Widyastuti as Director of the Research Center for Pharmaceutical Ingredient and Traditional Medicines to deliver the opening remarks. Prof. Yuli, the time is yours.
Bismillahirrahmanirrahim. Thank you, Ibu Marisa, for the time for me to give the opening remarks. Our honorable guest speaker, Professor Yoshimasa Tanaka and Dr. Henry Wasito, distinguished professor, dear colleagues and participants, ladies and gentlemen. Assalamualaikum warahmatullahi wabarakatuh and good morning. Indeed, it is my great pleasure to welcome all of you to joining today's talk. Actually, this is the serial webinar which are held by Research Center for Pharmaceutical Ingredient and Traditional Medicine. First of all, on behalf of Research Center for Pharmaceutical Ingredient and Traditional Medicine, I would like to express my sincere gratitude to Professor Tanaka from Nagasaki University, Japan, and also to Dr. Henry Wasito from Universitas Jenderal Sudirman, Purwokerto, as our guest speaker in this webinar. This research talk actually is a routine agenda of the Research Center for Pharmaceutical Ingredient and Traditional Medicines, which is held monthly and organized by every research group within the center. Today's talk was prepared and organized by Natural Product Chemistry Research Group that led by Dr. Abdi Wiraseptama, and kindly inform that today he will be the moderator in the next session. Today's talk take a theme related to the exploring and developing natural product chemistry for drug design and drug development. Ladies and gentlemen, exploring new chemical agents from natural resources for drug development actually is a real challenging, because the consequences of the global climate change in the near future, maybe it will lead to the new and re-emerging diseases, which is require new drugs for more effective therapy. Furthermore, currently, there are also many cases of drug resistance caused by abuse of drug, and this issue need to be anticipated with more intensive drug discovery research. Ladies and gentlemen, I'm pretty sure that this webinar will give us new perspective about the research experiences from Sensei Tanaka and Dr. Henry from their respective fields. Prof. Tanaka will share his research experiences dealing with marine-derived microbes and molecules for drug development. And I think it is very interesting topic, since Indonesia has blessed with the longest beaches area in the world, where all bioresources derived from there could be explored intensively and further studied for its potential in the future. Moreover, Dr. Henry will share the role of metabolomic approaches in pharmaceutical and life research science research. I wish you all the very productive webinar with exciting and encouraging discussion and also exchange of knowledge to build a prospective research collaboration, since the close and mutual collaboration is needed to share all resources so that research goal could be achieved more optimally. Ladies and gentlemen, last but not least, special appreciation goes to the committee, Dr. Abdi Wiraseptama and his colleagues who could organize this successful event. And please allow me to open this talk officially and hope you all to have an enjoyable webinar. Thank you. Wassalamualaikum warahmatullahi wabarakatuh. Time will be returned back to Mbak Marisa. Okay.
Thank you, Prof. Yuli, for the remarkable opening remarks. But ladies and gentlemen, before we head to the next agenda, we will have a group picture together. So please, our guest speaker and also all of the participants to open their camera so we can take the picture together. Okay. 1, 2, 3. One more. 1, 2, 3. Thank you. Okay, ladies and gentlemen, as head of the main agenda, we have a brief announcement regarding our research talk series, and we have many talks, research series, and this is our second research talk series. And the third research talk series will be held around on 24th of August. So please, all of this day participant could attend our next research talk series. Now we are moving to the main agenda today. This session will be hosted by Dr. Abdi Wiraseptama. Dr. Abdi Wiraseptama, PhD, is a head of the Natural Product Chemistry Research Group in the Research Center for Pharmaceutical Ingredient and Traditional Medicines. He finished his doctoral degree from Prin Songkhla University, Thailand, and since then have led numerous experiments regarding drug discovery for antimicrobial resistance and immunomodulator. He was also active publisher their team result and have 37 publications on high reputations journal index. So without further ado, I would like to invite Abdi Wiraseptama, PhD, to begin these sessions.
Assalamualaikum warahmatullahi wabarakatuh. Thank you so much, Dr. Marisa, for your introduction, for you introduce me. On this occasion, I'm going to be a moderator for the speakers session. So today, as we informed you, we have two good speaker. First is Sensei Yoshimasa Tanaka from Nagasaki University, and the second one is Dr. Henry Warsito from Universitas Jenderal Sudirman Purwokerto. So first, Professor Dr. Yoshimasa Tanaka will present his work around 40 minutes, and we will have a discussion around 10 or 15 minutes after every speaker finishes his talk. So before we start, I will introduce a little bit about Dr. Yoshimasa Tanaka. Prof. Yoshimasa Tanaka, thank you so much for joining us today. It's great pleasure and honor for us to have you here, and we hope you will share a good research work, because we know that you are one of the top researchers in the world, because I see in your CV you are one of Nobel Prize laureates in physiology medicine. So I will read Prof. Yoshimasa Tanaka VT. Okay. Prof. Dr. Yoshimasa Tanaka is present position and is the director and professor at Center on Medical Innovation at Nagasaki University. He got his PhD from Hokkaido University, have sort of research experience, and start from to be a research associate in Howard Hughes Medical Institute at Albert Einstein College of Medicine in New York, USA, and now he is the Professor and Director at Center on Medical Innovation, Nagasaki University. He has also very good research project. One of his research projects is development of cancer immunotherapy. Yeah, amazing, I think, because in collaboration with Professor Tasoku Honju, they got a Nobel Prize laureate of Physiology and Medicine in 2018. Wow, it's very. We are very lucky today. We will have good hearing with Prof. Yoshimasa Tanaka. So Prof. Yoshimasa Tanaka will today he's going to talk about marine-derived microbe and molecule for drug development, basis for supporting innovative drug discovery and life science research at Nagasaki University. Prof. Tanaka will share around 40 minutes, and after that we have the discussion around 15 minutes after Prof. Yoshimasa Tanaka finishes his presentation. Well, without further ado, I would like to invite Prof. Yoshimasa Tanaka to share, to give lecture to us. Prof. Yoshimasa Tanaka, time is yours.
Okay, thank you very much for your kind interaction. I'll share my slide. Okay, it's okay. Can you see my slide?
Okay. Can see the slide.
Okay,
Yes.
So today's talk is marine derived molecules and molecule for drug development. Okay, basis for supporting innovative drug discovery and life science research at Nagasaki University. Yeah, I'm a director and professor of the Center for Medical Innovation, Nagasaki University. Before I came to Nagasaki University, I had been working with Professor Honjo. So I went to Linderhof-Palles et al., Germany, about 4 years ago with Professor Honjo. And as you know, right after that, he was awarded Nobel Prize for Physiology or Medicine in 2018. And after the ceremony, he gave me a couple of Nobel medal shaped chocolates. Okay, today's talk consists of 5 parts: introduction and history of development of small molecule drugs and monoclonal antibody and marine microbial extracts and a nanobody. Okay, the first part is the introduction. So what is the impression of medicines? Okay, and the powder with bitter taste and tablets and capsules come up to my ordinary people's mind. But actually, this is a top 10 best-selling drugs in 2020. So the Humira, Keytruda, and Opdivo, these red ones are actually monoclonal antibodies. And then the black ones are so-called small molecule drugs. So 1, 2, 3, 4, 5, 6. So 6 out of 10 best-selling drugs are monoclonal antibodies right now. And then this is the worldwide sales of monoclonal antibodies. At 2000, the sales was almost nothing, but the amount of sales increased year by year. And then 2022, it reaches almost $200 billion. And then this is a rank 1 best-selling drug. And the Humira is an anti-TNF alpha monoclonal antibody drug. Okay. And then this is a rank 10. Actually, almost 20 years ago, we developed this drug. So Opdivo is an anti-PD-1 monoclonal antibody. Okay. This is a comparison of small molecule drugs and monoclonal antibodies. And then the small molecule drugs was produced around 120 years ago. So that means the scientists started to develop modern drugs about 120 years ago. Maybe I'll use the pointer. Okay, so the most small molecule drugs are derived from natural origin, so natural products or their derivatives, or organic chemists synthesize them. So for example, the aspirin and the penicillin or something like that. And then the most companies have developed this kind of small molecules until mid-1990s. And then after the late 1990s, the pharmaceutical companies tried to develop monoclonal antibodies. Okay? The monoclonal antibodies are produced based on molecular biology, immunology, and cell biology. And then now, the mainstream of the drug development is monoclonal antibodies. Okay, the second part, the history of the development of small molecule drugs. And then this is a cause-specific mortality rate in Japan between 1899 to 2019. And we don't have data during World War II. And then before World War II, most Japanese died of infectious diseases, including pneumonia, gastroenteritis, and tuberculosis. And then after World War II, the infectious disease declined rapidly, and instead, malignant neoplasms increased year by year. Okay, this is a simplified that picture. The infectious diseases declined rapidly due to the improvement of nutrition and public health and development of antibiotics. And then instead, malignant neoplasms increased year by year due to the prolongation of longevity. So it is imperative to develop effective treatments for malignancies now. And then about 100 years ago, many drugs were discovered by serendipity. Okay, serendipity means the occurrence and development of events by chance in happy or beneficial way. And this word serendipity was coined by Horace Walpole. One day he read the fairy tale, The Three Princes of Serendip. The Serendip is now called Sri Lanka. Okay, and then in this fairy tale, the heroes were always making discoveries by accident and sagacity of things they were not in quest of. So the Horace Walpole coined this kind of thing, the lucky, the serendipity. And then the modern drug development began after British Industrial Revolution. The Industrial Revolution was the transition to new manufacturing processes in the period from about 1760 to sometime between 1820 and 1840. This transition included going from hand production methods to machines. So before Industrial Revolution, people made things by hands, but after Industrial Revolution, they generate power and then they use machines to make things. Okay, and then during the Industrial Revolution, coal played a very important role. A great improved steam engine was designed and built in 1763 by James Watt. And then at that time, they just burned coal. But after that, gas fire was invented. The gas fire converts coal and water into carbon monoxide and hydrogen. So carbon monoxide and hydrogen. The resulting gas is the mixture of carbon monoxide and hydrogen is called syngas and is itself a fuel. And then after that, gas turbine and steam turbine were invented. A steam engine or steam turbine using syngas generates energy more efficiently than coal itself. So they can efficiently generate energy. But the problem was there are two byproducts during the process of making syngas. One is coke and another one is a coal tar. Coke is just a carbon, almost a pure carbon. So they just can burn coke. So it's okay. But the problem was coal tar. Coal tar was carbonaceous substances. So organic chemists try to convert coal tar something important or something useful. And then first organic chemists distilled coal tar and then benzene they obtain. And then when they treat the benzene with nitric acid, they obtained nitrobenzene. And then when they steam distill the nitrobenzene in the presence of tin as a catalyst, they obtained aniline. Aniline is basically a colorless compound, but the compounds containing aniline structure, so here, have colors, just like indigo. Indigo is a natural compound and derived from Indigofera saffruticosa. And this indigo is used to stain jeans, blue jeans. And then, and this is the British Empire. The East India Company drove the expansion of the British Empire in Asia at the time. But they had to overcome malaria when they colonized Southeast Asia, or they just occupied, colonized Southeast Asia. But at that time, quinine derived from the bark of a cinchona tree was effective for the treatment of patients with malaria. But the problem was that quinine is a natural product and very expensive. And then the organic chemists tried to synthesize quinine at the time. And then William Henry Perkin, 18 years old at the time, he tried to oxidize allyl-tridine to synthesize quinine. Okay, the structure of aniline and tridine is something like this. And he tried to synthesize quinine from aniline and the mixture of the aniline and the tridine. But he failed actually. And then instead, he succeeded in synthesizing this compound. And then this one was a purple dye. Okay. At the time, purple dye was very expensive. The natural purple dye was extracted from predatory snail. And then the structure is a 6,6' dibromo indigo. And then this one was very expensive. And then only high-ranking priests or kings could wear this kind of purple clothes. So that's why it is called royal purple. And then Perkin became very rich, actually, because of the synthesis of purple dye. And then many companies in Germany tried to synthesize dyes, actually, synthetic dyes. So orange, yellow, pink, or something like this. And then they stained the silk with these dyes. And silk is made of proteins. And then one day, the biologists thought, the cells are made of proteins. And if silks are stained with dye, maybe they can stain cells with dye. And then at the time, in Britain or in Europe, syphilis was the very big problem at the time. So many organic chemists tried to synthesize some compounds which can be used as drugs for syphilis. And then the Paul Ehrlich, he was a Nobel Prize winner in physiology of medicine in 1980. And he came up, a good idea came up to him actually at the time. And then if he can stain bacteria causing syphilis, and then that dye is toxic, maybe he can cure syphilis. Okay? And then at the time, the Japanese student was in his lab. So one of his colleagues, Hachiro Hata, screened the number of toxic dyes that had been synthesized in Professor Ehrlich's lab and found that acetophenamine or salvarsan could kill Treponema pallidum, which causes syphilis. And then Ehrlich later hypothesized that, and this is very important, by screening many compounds, a drug could be discovered and that would have antimicrobial activity, but not kill patients. This is the basis for the development of nearly all modern small molecule drugs. But actually, the salvarsan was too toxic, but it contains arsenic here. So this was not commercialized. And then the Gerhard Domark, and he's also the Nobel Prize winner in physiology of medicine in 1939, and he developed frontazil. And then this one is also the aniline containing compound, and then this is a dye, and then a little bit toxic to Streptococcus pyogenes. And he is credited with the discovery of sulfonamide glycoside. The first, and this compound is called frontazil, the first commercially available antibiotic. And at the time, accidentally, his daughter was infected with Streptococcus pyogenes. So his daughter was the first patient that treated with frontazil. And then this is a very famous story. And Alexander Fleming, he was also a Nobel laureate. When he cultured Streptococcus aureus, this plate was contaminated with penicillin-producing mold, and then he found the penicillin G. Okay, and then after that, pharmaceutical companies use a very big library set, and then they screened many compounds. And then the strategy is very simple. They first identify drug target, and then establish an assay system. They usually prepare 6 or 7 different assay systems, and then high throughput screening, and then hit to read compounds. So that this means the organic synthesis. And then after that, they select maybe 30 compounds or something like that. And then they check the ADME-Tox, the pharmacokinetics and pharmacodynamics. And then they choose usually one, and then perform clinical trials. And then whether or not the drug is efficacious to the diseases or not. Okay. And then the targets for the small molecule drug development is something like this. The most important thing is the receptors that recognize small molecule ligands. The original ligands are small molecules. That's why if they try to find the inhibitors, inhibitors can be a small molecule, so small antagonist. And another target is enzymes inside the cell. And then the enzymes recognize small molecule substrate. And then if they try to find the inhibitors of these enzymes, they usually find the small molecule inhibitor. So the target of the small molecule drugs are receptors or enzymes that recognize small molecule. Okay, the next part is a monoclonal antibody. And then every year, the journal Science, they select breaking through of the year. And in 2013, the cancer immunotherapy was selected. And cancer immunotherapy was initiated by William Coley. He was a surgeon in New York. And almost 130 years ago, one day, a cancer patient came to his office named Fred Stein. And then one day, he was accidentally infected with Streptococcus pyogenes. And he had a very high fever. And then he almost died at the time. And then he managed to recover. And then after recovery, interestingly, his tumor was gone. And then William Coley was very interested in this phenomenon. And then next, 1891 May, another cancer patient came to his office. And then William Coley injected living Streptococcus pyogenes to this patient. And then the tumor shrunk down. But the living Streptococcus pyogenes is very dangerous, actually. The patients may die because of this bacteria. And then when next cancer patients came to his office, he used killed Streptococcus pyogenes. So he injected the killed Streptococcus pyogenes to these cancer patients. And then tumor shrank down. And then after that, many immunologists and oncologists tried to develop cancer immunotherapy, including pathogen-associated molecular patterns, cytokines, tumor antigens, and dendritic cells, and tumor-specific T cells, and so on. But almost nothing worked, actually. And then about 20 years ago, almost all oncologists and immunologists gave up the development of cancer immunotherapy. But the cancer immunotherapy came up from the totally different field. That field, programmed cell death. This is the mouse fetus in the womb. In the early stage in the womb, the forefoot, the shape of the forefoot is something like this, something like fan. Okay? And then in the late stage, the forefoot becomes something like this. So during this time, the cells between fingers die. We call programmed cell death. In humans, our hands are almost the same. In the early stage in the womb, our hands are something like this. The shape is something like fan. But during the gestation, in the late stage of the womb, the cells between fingers die, and then our fingers become something like this. We call programmed cell death. So programmed cell death is very important. And then Professor Honcho tried to identify genes responsible for programmed cell death in the immune system. And then usually when T cells are stimulated very strongly, the T cells die. And then he tried to screen genes at the time. And then actually he identified genes. Before stimulation, this molecule is not expressed. But after very strong stimulation, this molecule is expressed. And then this one is called PD-1, programmed death 1. But right after that, many students in Professor Honcho's lab checked the function of PD-1, and then they found that PD-1 is not involved in programmed cell death. And then after that, the students and other staffs in Professor Honcho's lab tried to identify the real function of PD-1. And then they found out PD-1 is a receptor that transduces co-inhibitory signals or negative signals. So the T cells can recognize tumor cells, a virus-infected cells, something like this way. But if tumor cells or virus-infected cells produce PD-L1, and that interacts with PD-1, and then negative signal gets in, and then the T cells can no longer kill tumor cells or virus-infected cells. So this thing happens. So PD-1, PD-L1 interaction attenuates cellular cytotoxicity. But if we can inhibit the interaction between PD-1 and PD-L1, maybe we can inhibit the co-inhibitory signals, and then the T cell can kill tumor cells or virus-infected cells. But the protein-protein interaction, the area of the interaction is very wide. So we cannot use small molecules. So that's why we used anti-PD-L1 monoclonal antibodies or anti-PD-1 monoclonal antibodies. At the time, this one was the first experiment. So we first established P815 tumor cells expressing PD-L1, and we injected this PD-L1 expressing tumor cells into mice. And then in the mice, the tumor grew day by day, something like this. But if we treat the mice with anti-PD-L1 monoclonal antibodies, the tumor cells no longer grew. So this is the first indication that we can use anti-PD-L1 monoclonal antibodies or anti-PD-1 monoclonal antibodies, now called immune checkpoint inhibitors, to treat cancer. Okay, and then this is the expression of PD-L1 in ovarian cancer patients. And some patients express a very high level of PD-L1. And then some patients express a low level of PD-1. And then this is a survival curve after surgery. The patients expressing low level of PD-L1, they can survive longer, more than 10 years. And then patients expressing high level of PD-L1, the patients die. Only 40% patients survived more than 10 years. So that means that anti-PD-L1 monoclonal antibody is very promising to treat cancer. And then based on these findings, the biotech company, they're called the Medrex, they tried the critical trials. They treated the lung cancer patients with anti-PD-L1 monoclonal antibodies, and then the tumor shrunk down. And then they treat the lung cancer patients with anti-PD-1 monoclonal antibodies, the tumor shrunk down. So they found that even in humans, we can treat the cancer patients with anti-PD-L1 monoclonal antibodies or anti-PD-1 monoclonal antibodies. And then the company developed nivolumab or Opdivo. And then actually, this is chemotherapy, the docetaxel, and then this is nivolumab, Opdivo. So after lung surgery, the patients survived, about 15% survived for more than 2 years. But when we treat the patients with nivolumab or Opdivo, more than 30% patients survived more than 2 years. That's why the nivolumab Opdivo was filed. Okay, and then this is the picture. So this is the protein-protein interaction outside of the cells. So the target of the monoclonal antibody is the protein-protein interaction outside of the cells. Okay, so PD-1 is expressed outside of the cells, and PD-L1 is expressed outside of the cells, and then the target of the monoclonal antibody is here. So protein-protein interaction outside of the cells. So extracellular protein-protein interaction, or we can call extracellular PPI. So this is the mechanism. Okay, and then this is the comparison of the small molecule drugs and monoclonal antibodies, and who make or who develop drugs. Okay, in case of small molecule drugs, organic chemists develop small molecule drugs. But in case of monoclonal antibodies, the immunologists or molecular biologists develop drugs. And then this is the strategy to develop small molecule drugs. identification of drug targets, establishment of assay system, validation of the drug targets, and high throughput screening, and second screening or higher screening, and optimization of the drug, and other tasks, and PK, PD, and clinical trials, and finally, the drugs are on the market. So this is a kind of algorithm. Okay? But unfortunately, we have a limited number of drug targets. So the drug targets was the receptors on the cell surface or the enzymes inside the cells. We have actually in humans, we have only 20,000 genes in humans. So this kind of drug targets, maybe we have 2,000 or 3,000 or something like that. So after the development of 2,000 drugs or 3,000 drugs, we don't have drug targets anymore. So we moved to another drug target, that is a protein-protein interaction outside of the cells, and monoclonal antibodies can be developed. Okay? And then in this case, we have a problem actually. The monoclonal antibody are very specific. So if we develop monoclonal antibodies specific to humans, it doesn't work in animals. So that's why we have to test the efficacy of monoclonal antibodies in humans. So we call first in human clinical trials. So the university hospitals play a very important role to develop monoclonal antibodies. Okay. In the university hospitals, we can get POC, the proof of concept, in a small number of patients. And this process is relatively inexpensive. And then if we found if the drugs work very well, the pharmaceutical companies buy the university hospital derived ventures, or we call startups, or we call biotech companies. Okay. In the university hospitals, we can identify unmet medical needs, and then we can transfer unmet medical needs to the basic scientists labs. And then in the laboratory, we can develop drugs, and then we transfer drugs to the university hospital, and then they check the efficacy of the drugs. And then this is the new process to develop monoclonal antibodies. And then in the United States, many drug companies are involved, including Amgen and Genentech or something like that. But Genentech is a biotech company, but actually this is bigger than the Takeda pharmaceutical company. This is the biggest company in Japan. And then United States, there are many biotech companies or startups or venture companies. But this one happened after 1980s, actually. During 1970s, United States had a lot of problems, had a kind of Iranian revolution and oil crisis, Iran hostage crisis, and Chrysler on the verge of bankruptcy of something like that. And then the United States government looked at the innovation things, actually. So they thought innovation is very important. And then they checked the number of patents, that this is the intellectual property, and then they checked the number of patents between 1964 to 1974. And then they found that in the United States, there are enough patents, but the problem was the licensing, out-licensing. Out-licensing is only 4%. So they felt that maybe this one was the big problem. And then 1980s, and the Senator Bai and Senator Dole, and they submitted an act we call Patent and Trademark Law Amendments Acts. Okay, and then this was adopted in 1980s. A key change made by Dole Act was in the procedures by which federal contractors that acquired ownership of invention made with federal funding could retain that ownership. That means we usually use federal money to develop something, and then we can file patents. And then that patent was originally government things, but this act says the professors or graduate students or students, if they have patents, that patent belongs to the professors or graduate students or students. And then after that, SBIR and STTR programs was filed. This one is this one in 1982, okay, 1982, 2 years after the Bayh-Dole Act, okay. And then the government encouraged the foundation of the venture company or startups or biotech company in the university, okay. And then this is San Francisco, called Silicon Valley, or usually American people say Bay Area. Many biotech companies were founded at the time. And then, for example, the Genentech, they developed insulin, rituxan, Herceptin, Avastin, Actemra, many more antibodies of biologics. This is the situation in the United States, okay? And then, even after that, okay, this is a target of monoclonal antibodies, the protein-protein interaction outside of the cell. The number of this kind of target is also limited, actually. Maybe we have 1,000 or 200, 2,000 or something like that. So after developing 1,000 or 2,000 monoclonal antibodies, we don't have drug targets anymore, okay? And then maybe 3 years later, we do not have drug targets, something like this. And then what is the next generation drugs? So one possibility is the marine microbial extracts derived mid-sized molecules or nanobodies, okay? And the fourth part is the marine microbial extracts, okay? And then the next drug target is the protein-protein interaction inside of the cell, not outside of the cells, okay? And then the monoclonal antibodies is too big to pass through the membrane, so they cannot reach to this site, okay? And then a small molecule is too small to inhibit the protein-protein interaction. So maybe the next drug targets is the protein-protein interaction inside the cells, and drugs can be middle-sized molecule or nanobodies, okay? And then I talk about the middle-sized molecules. And a middle-sized molecule is produced by many organisms, including marine organisms. Okay. Nagasaki Prefecture is surrounded by seas, and we have many islands and gulfs and bays or something like that. So that means the biodiversity, we have a lot of biodiversity. And then we collected marine microbes from all over Nagasaki Prefecture. Maybe you can do in Indonesia the same thing also. And then this is the Tsushima Island. And then we went to Tsushima Island and collected many marine microbes. Okay, and also we collected sea urchin, crabs, shrimps, and sea hare, sea anemone, or oysters or something like that. And then we isolated marine microbes, something like this way. And then we grew marine microbes. And then also we collected sea anemone or sea hare or something like that. And we extracted small molecules and also the middle-sized molecules from these animals also. And then we established the middle-sized molecule library, and then we use these kind of molecules for screening. And then the last part is the nanobody. Okay. Nanobody is the next generation antibody. This is a small size monoclonal antibody. Okay. This is derived from alpaca and shark, or accurately, camelids and shark. Okay. The alpaca is something like this, as you know, and the shark is something like this. Okay. First, I'll talk about alpaca. Okay. And then currently, many monoclonal antibodies are available right now. So in the 1995 to 2025, okay, 3 years later. After that, we cannot develop this kind of ordinary monoclonal antibodies. And then next 10 years, maybe single chain antibody and bispecific antibodies can be developed. And then after that, we have to develop nanobodies. And then this is a structure of ordinary humanized antibody, a monoclonal antibody, or human antibodies. Okay, we have 2 light chains here and here, and we have 2 heavy chains here and here. And then in the monoclonal antibodies, use this site to recognize antigen. Okay, so to recognize antigen, only this part is very important. So that's why we just take out this part and then connect this one and this one. And we call this one single chain antibody. Okay, this is a next generation antibody. And then we can connect this single chain antibody, so two single chain antibody with different specificity. So for example, one is for CD3 and one is for TCR or something like this. We call this one bispecific antibody. Okay. But the next generation antibody is the nanobody. Okay. The story began in Brussels Free University in Belgium. Okay. At the time, the students have to analyze the human antibodies in the course. Okay. But at the time, in the middle 1980s, the HIV was discovered. So AIDS. Okay. And then the students are very afraid of dealing with human samples. Okay. And then students decided to use other animals. And one professor had the camel serum. And the student decided to analyze camel serum. And then they found very interesting thing. Most animals, including humans, monkeys, mouse, other animals, all animals have this kind of mold antibodies. Okay, this is a human IgG antibody, immunoglobulin G antibody. Okay, but the camel have different type of antibodies. It's very rare, very strange. Okay, there are no light chain. Okay, and then they have only heavy chain. Okay, a little bit shorter, but this is a heavy chain. So we call heavy chain only antibody. So camel have heavy chain only antibody. And then when this antibody heavy chain antibody recognize antigen, they use only this part. Okay, this one consists of only one domain, VHH. So this is a variable region of the heavy chain only antibody. So we call VHH. Okay, and then the camelids were evolved in the North America, maybe near Arizona or something like that. And then some parts of camel's ancestors went north to Alaska and Siberia to China. Okay, and some went to Africa. Okay, and they have one hump. They are called dromedary camel. And then two humps, they are Bactrian camels. And then some camel ancestors went down to South America, including llama, vicuña, alpaca, and guanaco. Okay, now we have 6 kinds of camels: llama, guanaco, vicuña, alpaca, Bactrian camel, and Arabian camel. And then Bactrian camel and Arabian camels are too big as experimental animals. And then llama and alpaca were domesticated. So that's why the scientists decided to use llama and alpaca to develop nanobodies. Okay, and then if we take only this part, antigen recognition part, we can produce this VHH domain in E. coli. Okay, and we can call this one nanobody. Okay, nanobody is characterized by these features: long CDR3 regions. CDR3 means a complementarity determining region 3. So this region binds to antigen. So this is long. So that means nanobody can recognize many kinds of antigens. And the physical chemical is stable. So because the nanobody is composed of only one domain, this one is very stable. Even at the temperature of 70 degrees Celsius or 80 degrees Celsius, this is still active. And the affinity is very high because it has long CDR3. We can select the high affinity nanobodies. And also the production of E. coli. This is very important because the monoclonal antibodies are very expensive because we have to use CHO cells, Chinese hamster ovary cells. That's why to grow CHO cells is very expensive. But if we can use E. coli, we can produce nanobodies inexpensively. Okay. And then the genetic modification, because the structure is very simple, we can modify these nanobodies very easily. Okay, but still alpacas and llama are pretty big. So that's why in Nagasaki University, we decided to use shark. Okay. The shark antibody, shark also has the heavy chain only antibody. The structure is something like this. This is a little bit longer than the camel heavy chain only antibody, but the antigen recognition site is the same here. Okay, so this one is called VNAR. NAR means the new antigen receptor. So the V is the variable region. So variable region of the new antigen receptor, VNAR. Okay, and then if we take out these genes and then produce this part in E. coli, we can prepare nanobody. In Nagasaki University, we have the Department of Fisheries. So we have ship to catch shark. This is the logo of our university. And we have a fish farming system in our university. And then this is a fish farming system. And this is a shark farming system. Okay. And then this is this white thing, the squid. We can give this one to the sharks. And then we can, the farm shark, this is the nurse shark, or called bullhead shark, because the head looks like bull. Okay. And then this is a nanuka shark, and then relatively small one. Okay, this is a nurse shark. This is the different name is the bullhead shark. Okay. This is a small shark, and we can take blood. You can see the blood here. It's very easy. This is the nanuka shark. This is me. So I tried to at the time tried to take bloods. So it's very easy to take bloods from shark, and we can prepare a nanobody antibodies. So this is the next generation antibodies. And then the middle-sized molecules derived from marine microbe extracts and nanobodies derived from shark, both are marine products. We can use these things as the next generation drugs. Okay, so today I talked about the introduction, the history of development of small molecule drugs, monoclonal antibodies, marine microbe extracts. This is the mid-sized molecules as the next generation drugs, and nanobodies as the next generation drugs. Okay. Thank you very much for your attention. This is all my talk. Okay.
Okay, thank you so much, Tanaka Sensei, for your nice presentation. I think it's very interesting for us to know the development and discovery of drug from alternative source. I think if all the participants have questions, you can straight away ask Tanaka Sensei. You can raise your hand, or you can drop your question in the chat room, and I will read the question and we will discuss later on. There is no question. Is there any question from the participant? Yeah, we have one question, Prof, from Nurfianto Gier. Is it possible to extract, the question is it possible to extract metabolite from plant and animal that proven to cure disease?
Okay.
Yeah, you can.
Okay. Because Nagasaki University, we use the marine products or animals or something like that. But of course, you can use plants and animals, the land animals also, because maybe Indonesia, the biodiversity is much higher, much better than Japan. So maybe we can use the plants and animals in Indonesia. So we can maybe collaborate together.
Okay. Any question? Prof, I want to ask you regarding the cancer immunotherapy that you mentioned in your presentation. I want to confirm about the PD-1. Is it the sort of an antigen or protein is expressed in all kind of cancer cell, or just a certain cancer cells?
Yeah, the cancer cells usually express very strange proteins. Okay, that can be an antigen. We call the tumor antigens. And then usually the CD8 positive, we call killer T cells. The killer T cells recognize tumor antigen, but that they kill tumor cells. But if tumor cells express PD-L1, because the T cells express PD-1, that PD-1 and PD-L1 interacts together, and then the PD-1 transduce negative signals in T cells. So in cancer patients, usually the T cells cannot kill tumor cells. But if we can inhibit the interaction between PD-1 and PD-L1, we can inhibit the transduction of the negative signal. and then the T cells can kill tumor cells. But the problem is, in Japan, we use PD-1 monoclonal antibodies or PD-L1 monoclonal antibodies to inhibit the interaction between PD-1 and PD-L1, but that is very expensive, actually. So that's why it's very difficult to use in developing countries. But if we can develop anti-PD-L1 nanobodies or anti-PD-1 PD-L1 nanobodies, that can inhibit the interaction between PD-1 and PD-L1. And because we can produce the nanobodies in E. coli, we can prepare the nanobodies very inexpensively. Maybe $20 or $30 are possible. So if so, maybe you can use nanobody anti-PD-1 or nanobody anti-PD-L1 to cure cancer in developing countries also. And also, for COVID-19, if we use the monoclonal antibody cocktails, we can cure COVID-19. It's possible, okay. If we mix 2 to 3 monoclonal antibodies, we can cure, we can treat the patients. But those 2 to 3 monoclonal antibodies are very expensive. But if we can develop nanobodies, maybe we can use that kind of things in developing countries also. So the next generation drugs are very inexpensive, but very efficacious, I hope.
One more question from me. I do interest about the what we call the cancer stem cell. So this what we call it, the PD-1 inhibitor can be used to how to say, to kill the cancer stem cell. How do you think, Prof? Because you know that cancers
it depends on the expression of PD-L1.
Okay.
If cancer stem cells express PD-L1, we can use anti-PD-L1 or anti-PD-1 monoclonal antibodies. Yeah, okay. It depends on the expression of PD-L1 on cancer stem cells. You can check the expression of PD-L1.
Okay, we have one question from our director. From my opinion, Nano Body seems to be a smart solution for the future medicine. What health problem can be treated with nanobodies can one day replace various types of therapy, not only drug, but also surgery. Your comment, Prof?
Yeah, that's possible. But right now, the current situation, the strategy or method to prepare nanobodies efficiently is not established yet. So because we have to farm sharks a lot, we need a lot of sharks. So, but in Japan, we have sharks, but maybe we need different kinds of sharks also. And then we have to select the very good shark to develop nanobodies. Maybe in Indonesia, you have also many different kinds of sharks, maybe. So maybe if possible, I'd like to try that kind of sharks in Indonesia, to develop more nanobodies. Because the kinds of sharks are very limited in Japan. So we want to use different kinds of sharks to develop nanobodies more efficiently. Maybe we need middle-sized shark.
Okay, thank you.
Okay, one more question, Prof. Joko Triwono, Prof. I would like to ask you about the potential of marine microbe extract to treat infection of antimicrobial resistant pathogen. Do you have any experience with that? Is there any specific general become a point of interest in your research?
Yeah. Many Japanese scientists right now are using our marine microbe extracts to develop drugs, something like that, actually. But maybe in Indonesia, your biodiversity is very excellent. So that's why maybe if we can prepare the soil microorganisms or plants or something like that, maybe we can also develop middle-sized molecules or something like that, I hope.
Any more question? Because Prof. Tanaka will have any meeting, right, Prof? Around. Okay, Prof, I would like to ask you about is this only use shark or we can use another type of fish, something like catfish that we can find easily in Indonesia? Sir?
Yeah, actually, only camels and sharks and rays have this kind of heavy chain antibodies. So if you have maybe rays, maybe you can use that kind of thing. Ray.
Okay, any more question? Oh, you can, okay, one more question from Afrianil. My content is important in marine biodiversity and ecosystem for socio-economic system. What do you think about the economic costs which is associated with the marine biodiversity due to high exploitation for medicinal?
Okay. Actually, to develop, for example, the nanobodies or nanobodies, we initially use sharks. Okay, but maybe we need for one antigen, maybe we use maybe 10 sharks or something like that. After that, when we produce nanobodies, we use E. coli, so we don't use shark anymore. So that's why we don't have to worry about the ecosystems.
Okay, maybe the Dr. Ibu Maratu Soleha can ask the question straight away to Prof. Tanaka.
Yeah, thank you, Pak Abdi Wira. Prof, I would like to ask regarding your the experience of antibody. Do you screening the antibody by how do you do that screening one by one, or is there any method in screening so we don't need to do much experiment?
Yeah, actually one by one. Usually we prepare 2,000 or 3,000 different hybridomers, and we check each one by one using the facts. It's a very laborious work, but it works.
Yeah. So you challenge in the culture of antibody.
Yeah.
The culture.
Yeah, that's true. Yeah, we usually use ELISA or FACS to screen monoclonal antibodies.
And then to connect within the molecule one and the other, how do you connect both of them? The linkers.
Linkers?
You for the connecting or
this is a strategy. Okay, so first we select 2 to 3 monoclonal antibodies after screening, and then after that we sequence the genes of the monoclonal antibodies. Okay, and then we mutated the CDR3 regions, and then we select much high the monoclonal antibodies with higher affinity, and then that's how artificially we select the high affinity monoclonal antibodies after that. Okay, usually we immunize animals, and then after that we take spleens, or we just take blood, and then we fuse the B cells and SP2O myeloma cell lines. Okay, and then after that we select 2 to 3 hybridomers, and then we sequence the genes of the antibodies, and then we mutate it artificially, and then we obtain the high affinity monoclonal antibodies.
More laborious work, yeah, Professor. Thank you for your answer.
Okay, thank you very much.
Any more question? I saw just now there is one raised hand, but disappeared suddenly. Okay. If no more question, maybe we can finish our first session discussion with Prof. Yoshimasa Tanaka. Thank you so much,
very
Prof.
much.
It's clear.
Thank you very much
For us to have you here, and very nice sharing today. And we hope we have any collaboration and for the near future with you. Thank you so much.
I'm happy to collaborate with the Indonesian people, researchers. Okay, thank you very much.
Thank you so much, Prof. Let me move a little topic to the different topic today. We have Pak Henry Wasito. He is our second guest speaker today. He's my colleague actually. Okay, before we begin before we proceed our next agenda, I would like to introduce the second guest speaker, Dr. Henry Wasito. Dr. Henry, how are you?
Okay, I'm fine. So it's okay.
So you will present in English, right?
Okay, I prepare my presentation in English.
Okay. Okay. Dr. Henry Wasito finished his B.Pharm, Bachelor of Pharmacy from Gajah Mada University, and got his Master of Science in Pharmacy in field of pharmaceutical chemistry and pharmaceutical phytochemistry from Mahidol University, Bangkok, and complete his PhD from Institute of Nautical Chemistry, Department of Chemistry, University of Natural Resource and Life Science, BRIN. His work in metabolomics, his research work targeted on non-targeted metabolomics with separation sciences and accurate mass spectrometry in biotechnology. He also helped present some publication and conference contributions. His latest publications is about metabolomic, I think, the alternative in source fragmentation with single-stage high-resolution mass spectrometry with high association confidence in non-targeted metabolomic. So today, Dr. Henry Wasito is talking about his field, the metabolomic LCMS related. So without further ado, I would like to invite Dr. Henry Wasito to give his presentations. Time is yours.
Okay, thank you, Mr. Abdiwira Setama, as moderator today. And I would like to share my screen. Okay. You can see my screen?
You can see.
Okay. First of all, I would like to say thank you for Professor Julie Wijastuti as Director for Research Center, and also Professor Yoshimasa Tanaka for super nice presentation regarding marine microbiomes for pharmaceutical research. And also thank you for organizing committee for giving me the opportunity today. And today I would like to present my experience. This is just short sharing regarding liquid chromatography and mass spectrometry for metabolomics. Actually, it can be applied for pharmaceutical and life sciences research. Before I start my presentation, I would like to introduce my previous research in Baku. Baku is University for Bodenkultur, Vienna, Austria. And my PhD, during my PhD, I worked with Professor Stephen Hahn. He is a head of the research group of analytical chemistry and metabolomic research. And in this university have a lot of instrumentation, sophisticated instrumentation actually for metabolomic, for small molecule, but also not only for the MS, but also have many instrumentation that for research in Baku. And the outline today, I would like to give a short introduction regarding metabolomic platform, including the LCMS. And after that, I would like to talk regarding LCMS targeted metabolomic and non-targeted metabolomics. And finally, I would like to conclude my presentation. As we know, metabolomic is defined as a comprehensive assessment of a large number of small molecules. Actually, the molecular weight is less than 1.5 kilodalton that present in different biological sample for better understanding of the biological system. Metabolomic is expanding rapidly with a wide range of application, such as identification of novel biomarker, and that correlated to certain disease, and assisting the enhanced development of precision medicine in the context of drug discovery investigating metabolomic pathway in order to better understanding of cellular processes, evaluating of cellular response mechanism to a variety of environment perturbation, and maybe there is a lot of other application. And metabolomic platform in the field of analytical metabolomic, there are two main strategies that are currently being used: targeted and non-targeted approaches. Targeted metabolomic allow the accurate quantification of analysis of known metabolite of interest. In comparison, non-targeted metabolomic assessing and detecting as many distinct metabolomics as possible. Understanding of cellular processes using metabolomic alone is actually insufficient to advance of comprehensive view of all biological process involved. Moreover, multidisciplinary approaches in combination with analytical chemistry, along with biostatistics, bioinformatics, biochemistry, are needed to assess metabolomic in biological sample. And therefore, combining metabolomic with other genomic, transcriptomic, proteomic approaches also required to achieve much deeper insight. Although integrating different multi-level omics data is still challenging. And now I would like to give some short introduction regarding the LCMS. As we know, a reliable analytical analysis of the metabolic of interest with high coverage is needed for metabolomic. Therefore, approaches analytical method are typically used to determine the metabolomic in the biological sample as comprehensive as possible. And LCMS. LCMS is an analytical technique that involves physical separation using liquid chromatography of target compound or analytes, followed by their mass detection by MS. Its sensitivity, selectivity, and accuracy have made it a technique of choice for detecting small molecules in metabolomic study. Moreover, no single analytical platform can perform a complete and extensive analysis of all metabolites in the biological sample in terms of identification and quantification. Therefore, multiple analytical technique, platform, and substantial instrumentation improvement are employed for targeted and non-targeted metabolomic study. The ability to resolve and quantify ion in biological sample after their conversion to gas phase, ion by MS offer the broadest platform and powerful analytical instrument for metabolomic study. And as we know, MS instrument comprises three main parts: ion source, and then with several ion optic, mass analyzer, and detector. Several ion sources are currently applied in LCMS, and electrospray ionization is one of the most prominent ionization sources for routine LCMS. Electrospray ionization is among the most use electrical energy to ionize molecule at atmospheric pressure from liquid solution and into a gas phase before transferring to the mass analyzer. Although electrospray ionization is a soft ionization technique, under some experimental conditions, isotope fragmentation can occur, creating challenges in terms of sensitivity and especially for accurate annotation of small molecule in non-target analysis, and potentially generating misannotation or identification of important metabolomic from molecular feature. And other challenge for electrospray ionization for metabolomics, such as one metabolite will yield several detectable ions, such as a primary ion, monoisotopic ions, isotopolog of primary ions, adapt, and corresponding isotopolog, and others molecular feature. So the approaches to deal with this problem, we can use the optimization strategy regarding ion source for the optimized condition, and also we develop acquisition and data processing strategy to aim to reassociate primary ion species in final dataset. And this is the mass analyzer. It this is the most prominent use for the mass analyzer. This is QTOF, and this is Orbitrap. Yeah, the most important part of the MS instrument is the mass analyzer, in which the ions are differentiated and separated based on their mass to charge ratio before being transferred to the detector. And appropriate selection of mass analyzer depend on the mass range, resolution, scan rate, detection limit, and application. Targeted analysis require high selectivity and high dynamic range mass analyzer, such as MSMS or triple quadruple or tandem platform with quadruple in combination with other mass analyzer. And moreover, non-targeted analysis need high accuracy, acquisition speed, and resolution platform like time of flight, MS, and Fourier transfer based using Orbitrap MS. And this is the workflow for targeted metabolomic. Metabolites are clearly defined in target mass spectrometry based to evaluate their variability, concentration, pathway, and flux in organism. And various methodology aspects should be considered in targeted metabolomic regarding chromatographic rotation time, specific ion transition, dynamic concentration range, polarity, collision energy, and others. And quantification assay for targeted metabolomic can be performed in using several acquisition strategies such as selected event monitoring, multiple reaction monitoring, and parallel reaction monitoring. And usually we use native and isotopolog label standard to confirm identity and accurately quantify metabolite in target metabolomic. And reference material also with full traceability meeting all metrological criteria play important role in this case. However, the lack of ability is still a concern, and especially for low-cost biological metric materials. This is my publication during my PhD. This study set out to comprehensive characterize an in-house reference material for an authentic biological matrix to improve harmonization and standardization in MS-based metabolomic. In this work, yeast extract derived from fully controlled and online monitored fermentation were assessed using Helix Orbitrap MS, and we also use open source software tools for data processing and evaluation. And targeted analysis approach was performed to evaluate homogeneity, stability, and metabolomic characteristic, and other parameters. And external calibration with full C13 label yeast extract also used as internal standard for quantification strategy. And this is some of the result. An in-house yeast-based material is promising as candidate for quality control reference material to support the demand of MS-based metabolomics. More than 70 metabolites, including amino acid derivative, nucleobase, nucleoside, and sugar and other small molecules, were comprehensively evaluated, in which most investigated metabolites are found to be suitable for reference material. And this is for non-targeted metabolomic workflow using LCMS base. And for non-targeted metabolomic workflow, actually the key point is how to define the high quality of molecular feature, because in non-targeted metabolomic workflow, first of all, we for the first time we should select the picking and then pick alignment, and after that we process the data, and after that we can confirm our finding with the library. We also can use the open source library for identification. But the key point for this strategy is how to find a high quality of molecular feature. And after that, we can analyze our data, our matrix data set using chemometric approaches such as PCA or maybe PLSTA and other chemometric strategy to define some finding that related to our goals in our research. And this is my publication regarding the non-targeted metabolomic. In this study, comprehensive non-targeted workflow is developed for single-stage high-resolution MS platform and several polar metabolomic from standard mixture solution were separated using HELIC. HELIC is hydrophilic liquid chromatography, and ASE, electrospray ionization TOF MS, determine their signal intensity in positive and negative polarity. And dataset processing in terms of picking, the convolution, alignment, and filtering for non-targeted workflow. We use non-targeted analysis open source software. Also, we use open source software, and identification of detected molecular feature was performed by comparing experimental mass spectra to the public spectral database. This is also open database. The finding for this publication contributing significantly to explain electrospray ionization parameter on in-short fragmentation and provide an alternative non-targeted analysis workflow using a single-stage high-resolution MS only to enhance the confidence of metabolomic identity confirmation for intracellular metabolomic study. And this is some of the result. The characteristic of molecular feature detection. After we filter the molecular feature after peak picking and peak alignment, we got from more than 500 molecular feature, and then after filtering, we got around 250 high quality of molecular feature, and some of them can be identified with a high score of confidence annotation, and the others still unknown metabolite, because as we know, the database for metabolomic, I think this is still bottleneck for the metabolomic study. And for the open source software, we use MS Dial, can deconfold of the MS spectra. For example, this is the extraction in chromatogram for histidine, and this is the raw data from the LCMS data. This is the MS spectra looks like this, and after deconfold it with the open source software, we can get the more clear MS spectra. And so if we compare with the library, it can enhance the score of the identification. And the characteristic of molecular feature detected in yeast in my for the application, I try to profiling of metabolomic in yeast, and more than 250 unique molecular feature were observed for H polarity after the feature filtering process, of which roughly 80 annotated metabolomic feature with the high score of annotation. And feature filtering was conducted to remove redundant feature via adapt ion, blank filtering, small peak shape and intensity correlated to across all sample, and more than 150 metabolomic in ethanolic yeast extract sample were reputatively identified, including amino acid, derivatives, sugars, sugar phosphate, and other small molecule. And I think that's all for my short sharing regarding the LCMS for metabolomic, and we can apply this method for other research topic actually, for finding the biomarker, and also for other metabolomic pathway and other application regarding the pharmaceutical and life sciences research. And finally, I would like to thank also for the Baku. There are Professor Stephen Han as a group head of the research group, and then for the Professor Gunda Kollensberger also from University BRIN, because in Austria we always collaborate with inter-university as well. I think that's all for my short presentation, and I will deliver to Mr. Abdi Wira.
Okay, thank you so much, Pak Henry Wasito, for your presentations about the metabolomic study based on LCMS, right? Liquid chromatography, mass spectrometry, best metabolomic for pharmaceutical and life science research. For those participants, if you have any question, you can ask Pak Henry Wasito straight away. You can use bahasa Indonesia and or English. Is there any question? Okay, there is one question from Fatimawali. What active compound can be analyzed by untargeted metabolomic? Your response.
Okay, I think all of the compounds, all of the small
I'm
molecule
sorry.
compound.
I'm sorry, Pak. Maksud saya target metabolism.
Oh, targeted.
Yes,
Targeted. It's depend on the methods that we use. Actually, when using LCMS, maybe it is better for the all metabolic, but for the volatile compounds, maybe it's better using GCMS because as we know, no single method can be performed to evaluate all metabolomic in cellular system. But for the targeted, actually, because for the targeted, we need the standard or reference standard. This is the limitation for the targeted analysis. And as we know, not all of the metabolomic have the standard or reference standard. But we can produce our standard. In this case, we use yeast to produce reference standard because, and also for the label standard, because in yeast, we can use the carbon source, actually C13 label standard in yeast, and then we produce our standard. So it's called with in-house reference standard.
Thank you.
Maybe one raise hand from Ratu Saleha. Please, you can ask straight away to Pak Henry.
Terima kasih Pak Abdi Wira atas kesempatannya. Pak, saya ingin bertanya terkait analisa kromatogram yang keluar dari LCMS. Itu bagaimana teknik menganalisanya? Karena saya biasanya yang kita lakukan adalah membandingkan dengan library ya. Tapi kadang-kadang ketika kita itu tingkat kepercayaannya berapa persen dengan library. Kadang-kadang senyawa yang bukan seharusnya ada di sana itu teridentifikasi gitu di tanaman kita gitu, Pak. Jadi agar hasilnya bisa dipercaya, bagaimana cara mengidentifikasi kromatogramnya?
Okay, I would like to answer this question with bahasa. Untuk mengidentifikasi metabolit, terutama tergantung ya, itu targeted atau non-targeted ya. Kalau dalam metabolomic itu istilahnya kita punya level identifikasi, level kepercayaan identifikasi. Ada level 1, 2, 3, dan 4, dan seterusnya. Kayaknya ada 4 ya secara umum. Level 1 adalah level yang paling paling tinggi. Karena apa? Ketika level 1 itu adalah kita membandingkan dengan standar. Ya, membandingkan dengan standar. Berarti sudah pasti kan dari retention time, kemudian dari profil MS-nya kita bandingkan dengan standar. Tentunya di sana tingkat keakurasinya cukup tinggi. Ya. Nah, untuk level 1, 2, 3, dan 4 itu istilahnya adalah putative annotation. Jadi kita hanya berdasarkan 2 parameter dari hasil MS. Tentunya misalkan 2 parameter dari hasil pemisahan LCMS. Misalkan adalah retention time dan M/Z. Ya, kalau misalkan kita punya data retention time. Tapi kan retention time kan tidak selamanya sama untuk setiap analisis ya, karena itu tergantung dari kondisi kromatografi kita. Maka dari itu berarti kita hanya tergantungi dari MS. Nah, ketika kita hanya menggunakan data M/Z itu tidak cukup. Maka kita bisa juga menggunakan data bagaimana profil isotopolognya. ya. Kemudian profil fragmentasinya, kalau misalnya kita menggunakan teknik ionisasi yang tinggi dengan hard ionization ya. Kemudian bagaimana tingkat kemiripan antara library ya, jumlah fragmennya yang terdapat di dalam MS spektra kita dan MS library. Memang kita bandingkan dengan library ya. Tetapi untuk membandingkannya itu tentunya akan ada skor di sana. Nah, di sini kalau untuk yang non targeted ya, kalau non targeted itu cukup unik dan ini masih belum cukup dipahami ya, terutama untuk yang biasa bekerja dengan LCMS juga gitu ya, karena saya beberapa nanyakan ya, ketika saya mengirimkan sampel misalkan dia nanya nanti datanya apa? Saya tidak butuh data PDF, saya tidak butuh data report. Yang saya butuhkan adalah raw data. Jadi benar-benar raw data. Nah, dari raw data itu nanti kita bisa ekstrak. Ya. Kita bisa ekstrak dari raw data itu dengan menggunakan software ya. Pada dasarnya kita di sini akan menggunakan teknik algoritma tertentu. Banyak software open source yang tersedia ya. Salah satunya misalkan MS Dial. Pertama tentunya kita harus picking dulu. Ya, kita tentukan di sana ada peak nggak, karena peak itu akan menggambarkan metabolomik. Meskipun peak itu juga tergantung dari proses pemisahan kita. Nah, kemudian setelah itu kita akan mendapatkan MS spektra dari peak yang kita miliki. Nah, caranya gimana? Kita bandingkan dengan library dengan melakukan scoring lebih dari satu parameter. Jangan hanya membandingkan M per Z. Meskipun itu data kita didapatkan dari high resolution MS, ya high resolution MS itu misalkan kita memiliki sampai 4 digit di belakang koma ya untuk molekular weight-nya gitu ya. Tetapi itu belum tentu menggambarkan senyawa kita. Ya, belum tentu menggambarkan senyawa kita. Nah, bahkan ya, ketika kita membandingkan dengan library, dia akan memberikan hit yang sangat bermacam-macam. Dan terkadang ada senyawa yang sebenarnya tidak ada di dalam, misalkan di tanaman ya, tapi
ya,
itu sering terjadi. Maka dari situ kita yang harus kita perhatikan adalah berapa skornya ya. Kalau dari hasil penelitian saya sebelumnya, karena saya juga membandingkan dari database dan sebagainya, ketika saya menggunakan MS Dial dengan skor total skor ya, total skor itu dia karena men-scoringnya MS Dial itu dari bermacam-macam teknik scoring ya, kemudian digabungkan jadi satu formulanya, itu ketika nilainya 70% itu tingkat kepercayaannya tingkat kesalahannya ya atau false negatifnya itu dari identifikasi itu hanya sekitar kurang dari 5%-an. Jadi memang kita harus bandingkan dengan library, kemudian kita bandingkan, makanya bandingkan juga terkadang isotopolog informasi itu juga cukup memberikan informasi. Isotopolog misalkan kan kalau dalam sebuah molekul kan pasti ada misalkan C aja ya, ada C12 dan C13. Tentunya di sana ada profil isotopolognya. Nah, kemudian kita bandingkan juga dengan parameter-parameter yang lainnya. Nah, karena terkadang kalau kita dapat data MS kan pasti sangat berantakan sekali ya, maka proses deconvolved ya, proses dekonvolusi itu menjadi sangat penting ya untuk mendapatkan spektra yang lebih jernih, maksudnya lebih jelas, karena noise dan sebagainya itu sudah dihilangkan ya dengan proses deconvolution itu.
Jadi itunya pembersihannya itu tahapannya apa tuh Pak? Maksudnya untuk deconvolution aja. Lalu raw datanya bisa masuk ke maksudnya di input ke open source ya Pak?
Ya, open source aja.
Open source software apa tuh Pak? Software Pak?
Kalau saya yang cukup familiar, saya menggunakan di sini softwarenya adalah MS Dial. Itu cukup karena cukup friendly user ya, dan ini cukup mudah. Jadi kalau misalkan non targeted itu dia dari mulai pemilihan picking sampai ke identifikasi bisa melakukan itu. Di samping juga masih banyak software yang lain ya, kayak metapo analysis dan sebagainya gitu. Karena software sangat banyak sekali yang open source juga bisa kita gunakan.
Itu satu-satu ya Pak ya? Satu-satu kromatogram atau sekaligus kalau ke MS Dial?
Kalau MS Dial itu satu raw data itu masukin aja ke sana.
Oh, satu-satu molekul kan? Bukan semuanya?
Enggak, satu raw data.
Semuanya berarti. Kalau misalnya satu tanaman itu kan banyak isinya, berarti semuanya aja dimasukin gitu.
Satu raw data. Jadi yang sering belum dipahami gitu ya oleh pengguna LCMS adalah karena pendekatan kita adalah non targeted. Ya. Kalau non targeted itu dasarnya bukan biasanya kan kalau kita menganalisis yang konvensional kan kita menganalisis biasa ini juga memang pendekatan non targeted juga ya, tetapi dengan menggunakan teknologi misalkan kita pilih 10 peak yang paling tinggi dari hasil kromatogramnya gitu kan ya. Kan biasanya seperti itu. Tapi dengan non-targeted ini sebenarnya kita tidak banyak teknis sebenarnya di sana ya. Ada SWOT, ada teknik all ions dan sebagainya ya. Kita pada dasarnya adalah kita hanya memasukkan raw data dari hasil pengukuran, tentunya yang scanning mode ya, bukan yang SIM mode atau mungkin MRM mode ya untuk LS untuk MS-nya ya. Kemudian kita analisis data di sana dari mulai peak picking, peak alignment, peak deconvolution sampai ke identification. Jadi kita ibaratkan itu data MS itu seperti sebuah lautan. Sebuah lautan tentunya di sana akan berisi, di sana ada kura-kura, kemudian ada ikan, ada kepiting, ada macam-macam lah gitu ya. Nah, MS data itu bisa diibaratkan seperti itu. Nah, kita ingin mengambil ikan yang ada di sana. Namanya ikan itu berarti ada sirip, kemudian ada ekornya, bentuknya seperti itu. Berarti dia akan mengkategorikan mana yang masuk ikan. Ikan itu akan dikumpulkan, oh berarti ada di sana ada 1000 jenis ikan. Nah, yang lain itu berarti hanya noise dan sebagainya gitu.
Oke, baik, gimana Bu Amaratu Soleha menjawab? Oke, kita ke pertanyaan berikutnya ya. Ada pertanyaan dari Anissa Elesetia. Saya menggunakan Waters untuk analisis LC-MS dan data yang diperoleh berupa raw. Saya ingin bertanya bagaimana untuk mengolah data tersebut dalam GNPS atau metabol analisisnya.
Oke, pakainya Waters ini LC-MS berarti menggunakan teknik bisa saja quadrupole dan sebagainya gitu ya. Untuk analisisnya bisa aja ya masukkan saja biasanya untuk pengalaman saya juga menggunakan kalau yang Agilent kan biasanya ada namanya Mas Hunter ya. Mas Hunter qualitative analysis ya. Itu software yang berbayar dari Agilent kan biasanya masing-masing vendor pasti akan memberikan softwarenya sendiri-sendiri ya, kayak Thermo itu ada Calibro dan sebagainya gitu ya. Nah, kalau untuk kita menggunakan data open source gitu ya, maka biasanya raw data itu harus kita konversi dulu, harus kita konversi dulu menjadi bentuk yang kompatibel dengan non-targeted software yang akan kita gunakan. Misalkan saya ingin menggunakan MS Dial. Dan dia akan kompatibel ketika bentuk raw datanya itu adalah dan akan lebih cepat mengolahnya adalah ketika raw data itu sudah dalam bentuk datasetnya adalah misalkan ABF format ya, ABF format. Maka kita harus convert dulu dari masing-masing data kita menjadi bentuk ABF format. Nah, di sini kita membutuhkan software converter pembantu lain ya, misalkan kayak MS Convert, kemudian ada ABF Convert dan sebagainya, sehingga nanti dari raw data yang dari Waters tersebut bisa kita konversikan menjadi bentuk raw data yang dikenali oleh open source dari MS Dial. Nah, dengan seperti itu nanti bisa dipilih parameter settingnya lebih lanjut untuk analisis sesuai dengan parameter setting yang kita tentukan. Intinya memang butuh raw data saja sih kita convert. Tentu kita pasti butuh converter di sana. Converter juga open source kok, jadi jangan khawatir.
Oke. Bagaimana Ibu Eva? Apakah menjawab dari pertanyaannya? Baik, kita masuk ke pertanyaan berikutnya dari Dimas Juaningsih. Izin bertanya, apa titik kritis dari pengujian yang telah Bapak lakukan? Di sebelah mana dan bagaimana pemecahannya?
Oke. Kalau untuk targeted analysis gitu ya. Kalau targeted analysis titik kuncinya adalah kita harus punya standar. Nah, itu. Dan terkadang standar itu sangat mahal sekali ya, dan belum tentu tersedia gitu ya, karena sangat sulit sekali. Itu kendala utama ketika kita ingin melakukan targeted analysis gitu ya. Dan juga kendala lainnya lagi di targeted analysis adalah terkadang metode yang kita gunakan itu tidak cukup untuk bisa menganalisis atau mengidentifikasi dari senyawa target kita. Misalkan saya menggunakan senyawa yang sangat polar. Nah, tentunya saya harus memilih jenis LC apa yang akan saya gunakan. Karena senyawa polar, berarti saya tidak bisa menggunakan reverse phase kolomnya. Jadi saya hanya bisa menggunakan berarti helix ya, hydrophilic liquid chromatography helix MS. Nah, ada juga strategi yang dikembangkan agar kita bisa bisa menganalisis dari ring metabolomik dari yang polar, sangat polar sampai ke yang non polar itu dengan menggabungkan kolom. Istilahnya adalah dua dimensi LC. Ya, jadi yang pertama kita menggunakan kolom helix, kemudian untuk yang selanjutnya kita menggunakan kolom pivot space. Nah, jadi digabungkan seperti itu akan memperluas coverage dari metabolit yang kita identifikasi. Nah, perlu diketahui juga ketika misalkan kita ingin targeted metabolomic itu tidak semua senyawa itu akan kedetek. Ya. Maksudnya akan kedetek, dia akan bisa dipisahkan. Sebenarnya kalau masalah dipisahkan tidak masalah kalau di MS ya, karena dia memiliki M/Z yang berbeda. Tetapi masalahnya adalah dia tidak bisa dipisahkan sehingga peak-nya itu tidak muncul. Jadi sebagai pengalaman, misalkan saya melakukan dari 148 standar metabolomik, mungkin yang hanya kedetek dengan metode helix hanya sekitar 20% atau 30%-nya dari 148. Karena biasanya salah satu cara paling gampang untuk melakukan targeted analysis ya dengan menggunakan scope yang berbeda adalah kita dengan menggunakan istilahnya adalah monster mix standar. Jadi monster mix standar itu sengaja kita gabungkan dari standar yang cukup banyak. Biasanya itu kita punya monster mix itu standar dari senyawa apa sampai senyawa apa ya. Nanti itu kan bisa kita simpan ya, kita freezing itu cukup dipakai bisa cukup lama sekali. Nanti ketika memakai cukup diencerkan saja. Nah, kemudian yang menjadi perhatian juga ketika targeted adalah linear ring. Linear ring itu juga patut menjadi perhatian. Jadi kadang-kala kan masing-masing metabolomik abundance-nya kan berbeda ya. Ada yang low abundance, ada yang high abundance. Mungkin kalau yang low abundance ini yang sering bermasalah juga gitu ya. Sehingga dia sulit tersamarkan antara noise dan peak. Itu kalau untuk yang targeted. Kalau untuk yang non targeted, kunci utama yang seperti saya sebutkan tadi adalah kuncinya adalah kita bagaimana cara mendapatkan high quality of molecular feature. Karena apa? Di LC-MS ya, tidak semua namanya peak itu menggambarkan satu senyawa ya. Tidak selamanya M/Z itu menggambarkan satu senyawa. Karena apa? Bisa jadi satu senyawa itu ketika kita analisis dengan LC-MS ya, misalkan aja dengan electrospray ionization, dia akan menghasilkan lebih dari satu nilai M/Z pada retention time yang sama. Bisa jadi itu karena fragmen ionnya, atau bisa jadi karena itu merupakan dimers-nya dari senyawa tersebut, atau itu merupakan adducts dari senyawa kita, misalkan M plus H, kemudian M plus NH3, M plus HCN, dan sebagainya. Jadi tidak selamanya M/Z itu menggambarkan satu senyawa. Bisa jadi itu adalah sebenarnya dari satu senyawa karena dia adduct-nya berbeda, atau mungkin dimernya, atau mungkin fragmennya. Nah, maka cara yang bisa kita lakukan adalah kita ingin mengumpulkan kira-kira M/Z tersebut masih dalam satu senyawa yang sama ya. Jadi tentunya di sini dibutuhkan algoritma tertentu, dan biasanya software non targeted analysis itu sudah bisa melakukan itu. Ya, istilahnya di sana adalah deconvolution dan juga menggabungkan sehingga dia bisa mengenali ini misalkan fragmen ion ini merupakan berasal dari rekursor ion yang ini. Itu dia bisa mengenali. Bahkan untuk senyawa yang coeluting itu pun dia bisa mengenali karena dia mengenalinya dengan algoritma tertentu ya, mana start peak, kemudian mana end peak, kemudian pada peaknya, kemudian dari bentuk peaknya sendiri. Nanti itu akan dihubungkan ya, dia kayak membuat semacam korelasi ya dengan nilai tertentu. Itu nanti software itu sudah melakukannya sendiri. Jadi seperti itu. Ya, kalau untuk terkait instrumennya biasanya yang kendalanya adalah di pompanya ya, vakum ya. Itu yang paling sering terjadi gitu. Pompa kadang-kadang trouble, kadang-kadang rewel gitu. Kalau pompa vakumnya, kalau vakumnya nggak bagus, ya kadang-kadang nanti hasilnya intensitasnya nggak bagus juga gitu.
Bagaimana Ibu Nimas Juangsih, apakah menjawab dari penjelasan Pak Henry barusan? Sekira menjawab ya. Kalau mungkin masih ada pertanyaan lagi dari partisipan. Bu Sofa, silakan.
Ya, terima kasih Pak Abdi. Sekali saya mengucapkan terima kasih banyak Pak Henry ya, sudah presentasinya sangat menarik mengenai metabolomik ini. Jadi kami di BRIN ini kita juga punya alat yang LC-MS yang Esikitov ya, dan itu mungkin bisa dipergunakan untuk metabolomik ini. Namun kami biasanya menggunakannya untuk penentuan MS saja gitu kan. Dan kita bandingkan library. Kalau misalnya metabolik ini, apakah di MS dial ini ada library-nya gitu? Atau misalnya kita dari yang PDF yang kita dapat, apakah bisa dimasukkan ke dalam tadi itu ya metabolomik gitu ya, seperti itu. Mungkin nanti ke depannya siapa tahu nanti mungkin Pak Abdi kita bisa mengadakan workshop ya atau training on-site atau mempraktekkan gitu ya mengenai metabolomik ini gitu, biar kami juga ada alat FTNMR juga ya, mungkin bisa nanti ke depannya dimanfaatkan untuk ke arah metabolomik ini. Kemudian juga kami di LSMS kan raw datanya itu merupakan MZML, apakah bisa bisa diinkludkan ke software, dimasukkan ke dalam software yang tadi Pak Henry sampaikan. Terima kasih.
Oke, ya.
Langsung aja Pak Henry.
Ya, yang pertama tadi terkait outputnya PDF ya. Tapi kalau menurut saya output dari sebuah analisis LCMS itu bukan PDF ya. Jadi raw data. Nah, nanti raw data itu setelah kita olah, karena misalkan masing-masing instrumen itu punya software sendiri untuk mengolah datanya ya. Kemudian nanti dari hasil pengolahan data itu baru nanti outputnya, layoutnya bisa dalam bentuk PDF atau bentuk teks dan sebagainya gitu ya. Nah, intinya yang kita butuhkan di sini terutama untuk analisis non targeted, sebenarnya targeted juga banyak softwarenya juga ya. Ada Skyline itu yang sering saya gunakan ya. Jadi dengan Skyline itu saya bisa menggunakan raw data. Raw data itu yang bentuknya kalau misalkan dari Agile itu .d. Jadi kayak satu folderan itu. Jadi satu folder jadi kayak yang benar-benar keluar dari alat itu yang ada di data itu yang kita gunakan satu folder itu. Misalkan kalau di Thermo itu bentuk filenya adalah .raw. Nah, jadi seperti itu. Jadi yang benar-benar satu folder belum diolah belum diapa-apain. Nah, itu yang kita butuhkan untuk analisis lebih lanjut. Nah, kemudian kalau kita sudah bisa dapat data itu, kemudian kan tinggal bagaimana cara kita mengekstrak data itu dengan menggunakan software. Nanti outputnya bisa macam-macam. Apakah bisa cuman bentuknya teks, misalkan karena kita hanya informasinya M/Z dan intensity dan sebagainya ya. Nah, kalau kita sudah dapat matriks tadi ya, nanti kita bisa analisis lebih lanjut dengan chemometric. Sehingga nanti istilahnya kita bisa menggunakan kayak PCA, kemudian PLSDA, kemudian hierarchy clustering, kemudian bisa pakai volcanoplot dan sebagainya. Itu tergantung dari tujuan kita ya. Kita ingin menyajikan data itu seperti apa sehingga data itu bermakna. Karena datanya kan hanya data matriks yang sangat banyak sekali. Jadi intinya apapun itu instrumennya itu bisa kita gunakan karena sudah ada software kayak MS Converter ya. MS Converter itu bisa mengkonvert semua dan bisa mengenali semua raw data dari vendor. Apapun itu, ya apapun itu bisa dikenali. Jadi tidak terbatas kepada vendor apa. Jadi kalau misalkan dengan cara seperti ini sehingga kita bisa makanya di sana kan seringnya kolaborasi ini ya, maksudnya harmonisasi kolaborasi sehingga ketika kita mengukur suatu sampel misalkan ke unit analisis tertentu, nanti data itu bisa kita saling pertukarkan raw data tersebut. Bahkan kalau kita lihat sebenarnya sudah ada database untuk raw data LCMS. Ya, itu sudah ada sendiri database yang karena memang sengaja ilmuwan internasional itu karena untuk raw data yang penting-penting misalkan cancer apa kan ya, itu kan sayang ya, raw datanya kan mereka sudah melakukan treatment misalkan apa, kemudian dianalisis dengan LCMS. Nah, raw datanya itu sudah ada disimpan di kayak semacam database gitulah ya. Kalau bisa di searching pakai Google ya. Itu bahkan kita bisa menggunakan data itu untuk diolah lebih lanjut. Ini biasanya yang banyak mengolah adalah orang-orang bioinformatik. Nah, seperti itu. Jadi raw data itu sangat bermanfaat sekali karena pengennya dunia internasional itu kan pengen raw data yang kita hasilkan itu sayang kalau tidak diolah, tidak dimanfaatkan lebih lanjut. Nah, maka dari itu bisa dimanfaatkan lebih lanjut. Kemudian tadi datanya sudah dikonvert ke MZ per ML format ya. Karena biasanya kalau .mzml ini adalah bentuk format raw data yang general. Bahkan cukup support untuk diaplikasikan ke berbagai macam data, misalkan ke metabo analisis, ke MS Dial, kemudian ke software-software yang lain itu cukup kompatibel kalau datanya .mzml gitu ya. Nah, ketika misalkan itu tidak kompatibel dengan software yang akan kita gunakan, maka kita bisa menggunakan konverter yang lain. Tapi kalau data .mzml ini cukup universal untuk beberapa software karena memang biasanya dikonvert ke MZML dulu.
Terima kasih Pak. Untuk softwarenya sendiri itu berbayar semua apa ada yang free untuk belajar gitu seperti itu?
Ya. Kalau yang berbayar ada, tapi kemarin waktu saya PhD karena saya memang sengaja sama supervisor saya bilang kalau bisa saya akan mengeksplor untuk semua software-software yang saya gunakan tidak berbayar. Ya, karena kan kita tahu sendiri kondisi di Indonesia, kalau kita disuruh berbayar ya kita akan kesulitan. Maka saya berusaha untuk yaudah berarti kamu ini aja, gimana caranya kamu mengeksplor software-software yang tidak berbayar, open source, itu bisa digunakan bahkan untuk library. Ya. Untuk library sendiri pun banyak open source ya, sebenarnya meskipun ada library kayak Matlin ya, Matlin itu sangat banyak sekali ya, tetapi kelemahannya kita tidak bisa mendownload isi dari folder Matlin itu, jadi kita hanya bisa memasukkan raw data kita, data misalkan prekursor sama fragmen ionnya, kemudian kita masukkan ke Matlin ya, Matlin itu kayak webnya ya, web database gitu ya. Nanti dia akan memberikan beberapa hit kira-kira ini metabolitnya apa ya. Tetapi kan kendalanya kita tidak bisa mendownload semua yang ada di Matlin. Tetapi kita pun bisa mendapatkan library free. Karena itu juga semangat dari para ilmuwan internasional, itu mereka yang terutama untuk standar-standar itu mereka memberikan free library itu. Kita bisa mengunjungi RIKEN misalkan, tulis aja RIKEN library, MS library ya, RIKEN ya, dari Jepang itu ya, karena dia yang mendevelop MS dial, itu kita akan dapat library di sana banyak sekali, baik itu LC, MS maupun ion mobility, bahkan ada gitu ya, ion mobility, MS data di sana juga ada. Dan yang cukup handal, cukup banyak itu adalah MONA database. Jadi kayak North American Metabolomic Database. Jadi MONA database itu juga dia memberikan, bahkan kita bisa mendownload ya, dalam bentuk formatnya itu adalah .msp. Nah, bentuknya .msp itu kita bisa download, ya memang downloadnya cukup gede ya ukurannya ya, karena library-nya cukup banyak ya, kadang-kadang sampai berapa gigabyte gitu ya, dan sebagainya. Nah, tapi itu bisa kita gunakan library itu, nanti kita tinggal connect-kan saja dengan MS dial tadi, nanti ketika dia ingin melakukan identifikasi dan membandingkan dengan library, dia akan mengelingkan langsung dengan library yang kita gunakan. Jadi kita tergantung, kita mau memilih library-nya mana. Ini seperti itu. Itu open source, gratis.
Mungkin nanti bisa di-share ya Pak, itunya software-nya yang open source ya.
Setuju Bu Sofa.
Gimana Mbak?
Setuju kalau bisa ada workshop.
Iya betul, nanti mudah-mudahan nanti Pak Abdi ya, kita nanti siapa tahu nextnya kita ini ya range ya untuk tadi itu untuk workshopnya ya. Ini enaknya sih sebenarnya onsite ya.
Mungkin terima kasih Mbak Sofa atas pertanyaannya. Ya, kebetulan juga saat kita ya dengan tim Pak Henry juga lagi develop ya metabolomik dari GCMS. Insya Allah hasilnya bagus ya Pak ya.
Datanya kemarin sudah keluar ya. Datanya sudah kita tinggal saya membuat reportnya.
Oke, insya Allah hasilnya bagus, nanti kita combine deh. Oke, dan mungkin nanti juga mungkin kita bisa arrange juga ya workshop on-site workshop, nanti kita bisa undang beberapa teman dari Pak Henry sebagai salah satu narasumbernya untuk metabolomics. Karena kita di sini alatnya cukup memadai ya, cuman itu dia kurang eksplorasi seperti itu. Oke, if any question, is there any question?
Mudah-mudahan dapat riset lagi ya Pak ya untuk yang tersangkut dengan metabolomics.
Oke.
Supaya bisa bekerja sama dengan BRIN dan bisa mengundang Pak Henry Wasito.
Ya, oke, baik, insya Allah Bu. Okay, I think enough. There is no more question from participant. Thank you so much for your participation. For myself, I apologize if there is some mistake or something during the technical error or something during this agenda, this presentation. And thank you so much, Pak Henry Wasito, for your participation and your sharing today. Hope to see you again, and we will build a collaboration in the near future. Okay, I think that's all. I return to our MC, Dr. Marisa Angelina. Assalamualaikum warahmatullahi wabarakatuh.
Thank you, Dr. Abdi, Dr. Henry Wasito, and also Dr. Professor Tanaka that have been leave. Yeah, maybe he has many
agenda
on busy. So we are very thank you to Dr. Abdi and Dr. Henry for insightful talk today and discussion. I believe many audience here gets much beneficial information regarding to the potency of microbe that report by Prof. Tanaka, and also metabolomic studies that report by Dr. Henry. We hope next we can have some collaborations, all of the participants in this talk. So before we are finished our event, we want to remind you about next research talks agenda that will be conducted on 24th of August 2022. And to close the event, we would like to say thank you for your active participation during these talks. And we have all of the participants enjoy our event today. And we are looking forward to you joining in our next talks. Thank you. Bye-bye. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam
warahmatullahi wabarakatuh.
Terima kasih Pak Henry. Jumpa.
Ya, terima kasih kembali ya, izin live.
Ya, Pak.
Assalamualaikum warahmatullahi wabarakatuh.
Ini dia berakhir ya.
Bukan, ini